Welcome. Good afternoon, everyone. Thank you for joining in this session of Wednesday afternoon lecture series. And it's a real pleasure and honor for me. My name is Anirban Banerjee. I'm in the intramural division of Ph.D. to introduce today's speaker, Harmit Malik, who comes to us from the Fred Hutchinson Cancer Research Center in Seattle, where he's a professor and co-associate director in the Division of Basic Sciences and an investigator of the Howard Hughes Medical Institute. Harmit studies the cause and consequences of genetic conflicts that take place between different genomes or between components of the same genome. And he's interested in understanding these, what he calls molecular arms races, and how they drive recurrent genetic innovation from the perspective of both evolutionary biology and human disease. Harmit and his colleagues have used an evolutionary lens to dissect and discover both primate antiviral as well as viral adaptation strategies, and this work has helped found the field of paleovirology. The other area of research in Harmit's lab is the study of rapid evolution in genes involved in essential cellular processes such as chromosome segregation and mitochondrial biology. Together with Steve Henikoff, he proposed the centromere drive model, which posits that unusual genetic conflicts during meiosis drive the unexpectedly rapid evolution of centromeric DNA and proteins. Harmit's entry into biological science research was atypical. He received his undergraduate degree in chemical engineering from the IIT Bombay, now called IIT Mumbai, and in his final year was interested in a new course titled An Introduction to Molecular Biology. His core requirements prevented him from actually taking that course for credit, but his teacher, Professor K.K. Rao, offered to teach him the course one-on-one in the afternoon, provided Harmit kept up with the readings. Harmit did that, and of course some more, because that, together with the simultaneous reading of The Selfish Gene by Richard Dawkins, spurred a deep interest in selfish transposable elements that led him to a Ph.D. in Thomas Eichbusch's lab at the University of Rochester, followed by a postdoc in Steve Henikoff's lab at the Fred Hutch, where he started his work on evolution of centromeres and centromeric proteins. He started his lab in 2003 at the Hutch and has stayed there ever since. He's received a wide number of awards. I'm only going to name a few: the Pew Scholar, the Searle Scholar. He was an Alfred P. Sloan Foundation Fellow. He was a recipient of the Presidential Early Career Award in Science and Engineering, NSF Career Award, the Eli Lilly and Company Elanco Research Award from the American Society of Microbiology. He's an HHMI investigator. And I want to specifically mention the Sastra Obaid Siddiqui Award for Excellence in Life Sciences, named after Obaid Siddiqui, the founding father of molecular biology research in India. He was inducted into the National Academy of Sciences and the American Academy of Arts and Sciences. Harmit is also very keen to promote inclusion in science and is active on social media, encouraging people to get involved in science and breaking down the barriers of entry into science. And when leading practitioners of science do this, they build role models and build community that is particularly inspiring for younger researchers. And for that, we have to be collectively thankful for folks like Harmit. Thank you for coming to the NIH, Harmit. Look forward to your seminar.
Thank you very much, Anirban. I feel a little bit cheated because I was assured there'd be no more than 20 people into the auditorium, and I'm pretty sure that that's not true. Okay, well, I was told that there are going to be coffee and cookies after the thing, so you just have to wait for about an hour. It is also the longest day of the year, and I hope to not make it seem even longer. So today I'm actually going to tell you about some of the work that we're doing in my lab on host antiviral proteins. I just want to advertise, if those of you are keen to get more of this, I am also giving a lecture in the NHGRI tomorrow, and you're all welcome to attend that as well. So before I get into the meat of today's talk, I just wanted to give you an introduction into what inspires all of the work that goes on in my lab, which is really about genetic conflicts. And the idea of genetic conflicts really emerges from a very unusual source, so I thought I'd spend a few minutes discussing that, which is the fictional character, the Red Queen, who was introduced to us by Lewis Carroll in his books about Alice's adventures in Wonderland, where at one point, in response to Alice's complaint, the Red Queen retorts that it takes all the running you can do in Wonderland to keep in the same place. Now, those of you who run labs can totally appreciate the red queen interaction as like a really important lesson for life. But unsurprisingly, this also was a very important lesson for evolutionary biology and led to the formulation of the red queen hypothesis, which I like to think about as the second sort of incarnation of Darwinian thinking in evolutionary biology. Whereas Darwin and Wallace had been very interested in the role of natural selection and how organisms adapt to their environments, they had predominantly focused on their adaptation to their abiotic environments, temperature, moisture conditions, et cetera. And Lee Van Balen and his peers really sort of explicitly pointed out that a large part of that environment is also made up of competing species, which are often competing for the same fitness space. So it was impossible, for example, to consider what was going to happen in the fitness of snow leopard population if we'd failed to consider what was happening in the snow hare population, because the adaptive strategies of one species directly tied in with the adaptive strategies of the other. The red queen hypothesis is a very important theorem. It actually applies from everything from ecology to public health. And what labs like mine are very interested in is adapting this inherently ecological theorem into molecular terms. So I'm going to make a very small leap between arms races that occur between prey and predators versus arms races that occur between antiviral proteins. And today's lecture is going to be exclusively on innate antiviral proteins and viral proteins. Since Anirban is my host, I'll just point out that in this high-resolution structure of an antiviral protein recognizing a viral protein by some sort of epitope recognition, it's going to encounter the viruses as soon as they enter the cell, extinguish the virus from the cell, eventually from the organism, and eventually from the species. This, of course, puts a lot of Darwinian pressure on viral populations to rapidly evolve to a new state that is no longer capable of being recognized or antagonized. And this can happen at the level of a single amino acid mutation, which, given the very rapid mutation rates afforded to viruses, can happen very quickly. Over evolutionary time, this will actually force the antiviral protein to now reestablish its antagonism via mutation. But this is a much slower step because we now need to rely on generational mutation time, which is, of course, much slower in species such as ours. So a question that often arises is, how is it possible to think about these as contemporaneous arms races where one party is so much more adept at evolution and mutation than the other? And the response is that if this was a one-to-one interaction, it would be game over just because of the inherent asymmetric nature of these arms races. But luckily for us, it's not a one-to-one interaction. In fact, we have nearly 200 antiviral proteins encoded and often triggered by the interferon system that basically are dedicated to the job of interrogating incoming pathogenic viruses and bacteria and basically expunging them. And really, for the host to win, it really only needs to win one of these interactions in order to do its job. Whereas for a virus, even a very rapidly evolving RNA virus, it needs to run the entire gauntlet of antiviral proteins provided, and that's where the math begins to add up. Antiviral proteins also don't encounter just one pathogenic viral variant. As we have all seen, we've already seen multiple pathogenic variants of the same virus multiple times. And if you use public transport, you probably encounter them on a weekly basis, or if you have kids in daycare. Those are the real front lines of infectious disease in our species. And so really you begin to appreciate that antiviral proteins and viral proteins are locked in this many-to-many interaction. But even if you were to focus on the focal one-to-one interaction, you'll appreciate that this is a relentless engine for forward evolution. And what drives this engine is the fact that one party is always on the losing side of this arms race, resulting in genetic innovation being constantly spurred by the nature of this interaction. So there's always going to be an evolutionary advantage to be gained by this innovation. And what labs like mine are trying to do in a nutshell is look backwards in the sequences of viral proteins and the sequences of antiviral proteins to try to reconstruct the steps that took place in the course of these arms races and understand their biochemical and their virological outcomes. One special type of innovation that is quite easy to imagine is focusing on protein coding gene innovation. And this is just a brief primer for those of you who don't think about this on a daily basis, where if you were to consider a hypothetical piece of a protein coding gene, which encodes for 3 codons and 3 amino acids, we can classify all of the nucleotide substitutions that occur here over evolutionary time into those that will not alter the amino acid being encoded. We call these silent or synonymous changes, versus those that instantly altered the protein coding gene because of the nature of their amino acid substitution. We call these non-synonymous or replacement changes. Now, normalized as DS and DN, we have different ways of viewing what are the evolutionary forces that have driven each of these changes. We make the simplifying assumption that silent or synonymous changes are really not visible to natural selection. We do so in spite of the fact we know that this assumption is wrong. There are many very good examples where synonymous changes can affect protein translation rates and even protein stability. But we nonetheless make this assumption because as a first order approximation, an average silent or synonymous change is much more benign than an average replacement change. So we view DS accumulation as a proxy for how many mutations have occurred, as a proxy of almost like a clock-like fashion. The longer time of divergence, the more kind of your DS is going to diverge. Whereas natural selection will dictate what will happen with the DN term. If you do not tolerate amino acid changes, you'll basically have a DN which is much less than what you'd expect. Whereas if you actually favor replacement changes, you'd have a DN that's much higher than what you expect. Those of you who are used to looking at protein alignments inherently use this tool, even though you might not be thinking of this as the DN-DS tool, because when your eye is drawn to those positions that are completely frozen in evolution, despite millions of years of divergence, let's say catalytic residues of an enzyme, what you're doing is making a mathematical calculation of the number of mutations that must have occurred that altered the amino acid at those critical positions, but all of those were purged because they were deleterious for function. In other words, we use this proxy of how selection has acted to purify the population of presumed deleterious amino acid changes, or a proxy of high evolutionary conservation for function. So if it's highly conserved in evolution, despite millions of years of divergence, it must be important to have essentially survived mutation. But we sometimes make the mistaken assumption that things that are not conserved in evolution must therefore not be important. In fact, my entire lab studies cases where the rate of amino acid change much exceeds what you'd expect by chance alone. And that's acting not because you suddenly become much more sensitive to DS-type changes, but that's occurring because selection is now acting positively to accelerate the rate of fixation of amino acid altering mutations because they were beneficial in the context that they occurred. And we would make the case today that just like high evolutionary conservation is a proxy for functional importance, this constant relentless signature of innovation at the amino acid level is also a proxy for functional importance, but this one is a lot more specific about genetic conflicts. The nice thing about having such a simple tool is we can use this to apply to all protein coding genes in the genome. We can do this in our species. We can do this in Drosophila species or whatever your favorite group of species is. In our own species, we see that the overwhelming signature is that of purifying selection. So once again, if the only effect was about mutation, then all of the genes would be piled up around this dotted line of DN-DS equals 1. But because most protein coding genes are basically in this leftward histogram, this gap between the dotted line and the histogram really indicates how natural selection has actually purged all of the amino acids that have probably taken, amino acid mutations that have taken place during the course of primate evolution, but were presumed deleterious and purged out, which is why we don't see them anymore. But even in this very whole genome crude sort of DN-DS average, you can see there is a blip of genes which is very much in the fast evolving class, which is breaking the speed limit, if you will, that is imposed by mutation. And unsurprisingly, immunity genes are heavily overrepresented in this category. And in particular, today's genes that I'm going to talk about, innate immune genes that defend against viruses and bacteria, are heavily overrepresented here. That's what I'm going to focus on for today's lecture. We are also really interested in categories of genes that don't make any sense to belong in the fast evolving class, but that's actually the topic for tomorrow's lecture. Returning to this sort of intuition about how to think about purifying selection and positive selection on a sort of biochemical basis, let's imagine an interaction between two host proteins whose interaction is really important, let's say for a signal transduction pathway. Now we can imagine that mutations will occur either in one protein or the other protein that might perturb this interaction, but that would be deleterious because you're essentially breaking the signal transduction pathway. And the likelihood that one mutation will encounter the other mutation, given that both are rare in the population, is exceedingly rare, which means that you will come back millions of years later and find that purifying selection has largely maintained the interaction interface because that is so important in the course of evolution. So this particular protein-protein interaction has basically been left unchanged by natural selection. In contrast, if you imagine what happens in a host virus arms race, here we basically bounce between two states, between the host winning versus the virus winning. And because what's in the benefit of the host is actually to the detriment of the virus and vice versa. So once again, we've got a protein-protein interaction interface, but this one cannot stay stable, and it's constantly chiseled away by adaptive evolution, either on the host side or the virus side. And moreover, you can actually, if you're even paying attention to this cartoon example, you can appreciate that we don't expect rapid evolution to be randomly distributed over these proteins, but in fact concentrated on those residues or domains that maximally affect the binding affinity between the host and protein interactions. So many years ago, armed with nothing more than this intuition, we wondered whether we could actually simply ask where positive selection occurs on the surface of the proteins as a first-order predictor of what the interaction surfaces between host and viral proteins might be like. So in other words, rather than focusing on the most conserved parts of proteins, like most molecular biologists, here we are actually focused on the least conserved parts of the protein in order to decipher what the specificity domains might be. Along the way, we've also realized that actually we learn a lot about susceptibility determinants when we do this. So there are many instances where one particular virus is highly pathogenic to one species, but a very closely related species is almost completely immune. And we've really sort of hypothesized in the early days of genomics, this must be because our immune gene repertoires are totally different. It actually turns out that immune gene repertoires are not totally different. They're basically almost the same, but the tuning of each of our immunity genes is different because we've each encountered a different private history of pathogenic viruses. And what's really spectacular is we can completely graft this tuning from one species into the other just by making single amino acid changes at the critical interaction interfaces. And that's what I'm going to show you today. We've done this for nearly a dozen antiviral proteins. They all have completely different mechanisms. Some of them are ubiquitin ligases, some of them are cytidine deaminases, some of them are protein kinases. They all work against completely different classes of viruses entirely. And yet, in each case, we can use this positive selection-driven approach to correctly predict the interaction surface and graft changes that will make the host move from a losing side to a winning side, which means that really the rate-limiting step to dictate the course of the virus infection in all of these cases is what is the binding affinity of the initial interactions between the viral and the immunity proteins, which is why this approach is actually so consequential. If you'd like to read a little bit more about this, here's a slightly out-of-date review in Annual Reviews of Genetics written by a former postdoc, Matt Doherty, in the lab. So today I'm going to present two vignettes about how we can sort of take these approaches and actually graft them to make even more insights into evolutionary landscapes. So the first protein I've chosen to talk to you about is a protein called Trim5 Alpha, because this is actually what I used to get into the business myself when I was starting my lab more than 2 decades ago now, almost. So Trim5 was actually discovered in a beautiful screen done by Joe Sadrowski's lab, who realized that old world monkeys like rhesus macaques cannot get infected with HIV-1, and this protection is actually at the cellular level. It turns out that when HIV-1 enters the cell and sheds its envelope at the cell surface, it enters as this proteinaceous capsid in the cytoplasm of the host cell, and Trim5 is actually a cytoplasmic sentinel, which basically is always expressed, always around, and it intercedes with the incoming retroviral capsid and forms a lattice-like structure leading to the rapid degradation of this retroviral capsid, preventing the virus from undergoing its otherwise normal life cycle of reverse transcription and integration into the host genome. The entire success of Trim5 is dictated by the avidity and kinetics of the interaction of the Trim5 protein to the capsid core. So the reason rhesus is so successful is because it has extremely high avidity for the HIV-1 capsid. We also have a Trim5, which is also very protective against many retroviruses, but our Trim5 version is not effective at correctly binding the HIV-1 capsid, which is why we do not have biologically meaningful protection afforded by the human version of Trim5. So many years ago, armed with this sort of paradigm about genetic conflicts and arms races, this time between Trim5 and the capsid gene of HIV-1, we thought we could actually predict what the interaction interface between Trim5 and capsid would be, again, by taking a look at where the rapid evolution in Trim5 might have taken place. And to our surprise, even though Trim5 is nearly 600 amino acids, almost all of the rapid evolution in Trim5 occurs in this very tight window, 11 amino acid stretch in humans, which is a 13 amino acid stretch in rhesus. So it's almost as if we basically focused all of the energy or all of these subsequent interactions down to just one domain of the V1 loop. As I pointed out earlier, human Trim5 confers only twofold protections against HIV-1 in cellular assays, whereas rhesus Trim5 encodes nearly 100-fold protection, which obviously is biologically protective. Interestingly, however, just taking the V1 loop, which remember is the most rapidly evolving part of Trim5, and swapping it from rhesus into human is able to confer nearly complete protection in the otherwise ineffective human Trim5 backbone. So this is one of those examples where you can actually see how rapid evolution can guide us to the interfaces where we can actually graft specificity from one backbone into the other. Even more spectacularly, just one amino acid change at one of these critical positions, this proline instead of an arginine, is able to confer what would be biologically meaningful protection in the human Trim5 backbone. Yes? This is the qualified unit of protection. I'll talk more about the assay in a few minutes, but this is just a fold protection relative to an empty vector control. Yeah. So this just basically provides the example that you can have single amino acid changes that could actually be meaningfully protective. I'll just preempt the question here that unfortunately, we do not have any standing variation in the V1 loop that would be protective against HIV-1 due to an adaptive sweep that went through Trim5 right at the out of Africa sort of migration of humans. We wiped out all polymorphisms and we've not recovered enough and imposed enough selection for that to be a protective landscape here. However, Trim5 is very polymorphic in old world monkeys. So this project actually started off with this very old study where we had shown that individual amino acids can actually be rapidly evolving and confer protection. But a new postdoc joined the lab and she was rereading all the old lab papers and she basically realized, well, you call this rapid evolution, but there's not that many changes if you actually think about them. It's almost as if evolution is moving within a box where only a few amino acid changes are permitted per position. And is that really a constraint? Or is that an instance where helpful mutations are really rare? Or is this just evolutionary happenstance where these are the mutations that happen to get fixed? And maybe there's a wide spectrum of mutations to choose from. Ironically, we don't have the means to distinguish between those possibilities because we are looking at all of the successful Trim5 variants, right? These are all variants that fought off something and have survived. So we cannot separate mutation from selection. So she decided that that's the project she wanted to do, mostly to prove my earlier discussion to be wrong. So this is Jeanette Tenthery's work that I'm going to describe today. So she really wanted to ask that if I were to liken the evolutionary landscape of Trim5 mutation in this XY axis as the sequence axis of Trim5 mutation, where the height of the peak determines how much the HIV-1 restriction is occurring, you can see that rhesus happens to be on one of the peaks and human Trim5 happens to be in one of the valleys. So she wanted to ask, are winning solutions just very rare to navigate in this fitness landscape? And if you occur on a winning solution, is that very mutationally fragile or is that mutationally resilient? So to do this, she needed to actually do an experiment where she would introduce all possible mutations and then subject them to selection. And we can do that thanks to nice advances in deep mutational scanning methods pioneered by Stan Fields and others. So in this case, we did not actually do a scan of the entire Trim5 because we were only interested in the V1 loop. So we introduced all possible single amino acid changes in the V1 loop of human Trim5. And we introduce all of these variants into cat cells. Now, the choice of cat cells is partly historical. This is the cell line that Joe Sadrowski had used to actually do his initial Trim5 experiments. But that turns out to be very fortuitous that he did that, because cats and dogs have independently lost their Trim5 for reasons we can actually discuss in the Q&A section, which means that all of these cat cells each has now an individual human Trim5 variant, each slightly different at the V1 loop. And that's all the Trim5 that they're capable of encoding. So we subjected this population of cat cells, each encoding a different human Trim5, to infection with HIV-1, but this HIV-1 has been engineered to only encode GFP upon integration. So it doesn't have the HIV-1 genome, it just has the ability to introduce GFP upon integration. For the aficionados, I should point out that we do this at a high multiplicity of infection because we want to maximize infection rates in this assay. So this assay is going to result in two consequences. Consequence number one is you're going to turn cells green. This means you were not able to prevent HIV-1 from integrating its genome in the cell. That means you did not have a protective Trim5 variant. We are interested in the non-green cells because for some reason they were protected against HIV-1, which is why the HIV was not able to integrate into the genome. So those could be encoding restrictor variants of Trim5, which is what we are interested in. But we are also convinced that there are probably some uninfected cells, which despite our best efforts, did not get infected with HIV-1, and those would be a source of noise, and we want to eliminate them. So Jeanette went through 2 rounds of infection where she sorted all of the GFP negative cells and subjected them to a second round of HIV-1 infection. Now it's very unlikely that a cell remains uninfected in both rounds. And simply by sequencing the Trim5 variants in the starting pool and the ending pool, she gets enrichment scores for how likely is it that you have a protective Trim5 allele variant. So it's a one-part experiment in which we have tested all possible Trim5 variants of V1 and assessed them in terms of their enrichment relative to the background. This is sort of just a sanity check here where we've done 2 biological replicates of this, and we find that the enrichment scores are beautifully correlated. And so that's, of course, very reassuring. A second sanity check came when we basically look at the enrichment scores on this waterfall plot, where the leftward you are, the more enriched, which means the more restricted you are. You'll notice that the wild-type human Trim5 is here in a cluster. There's actually more than one Trim5 variant, which are different at synonymous positions. They're basically identical because they encode the same protein. Even though I told you that human Trim5 is very poor against HIV-1, you'll notice that it is still statistically better than Trim5 variants that end up with stop codons in our mutation strategy, right? So this is like a very reassuring kind of step that we are making a biologically meaningful prediction about restriction. What about testing individual variants? This is actually in response to the question about the nature of the assay. So the nature of the assay is we basically infect cells with increasing viral doses, because essentially you're able to overcome whatever protection we have as long as you have enough of a viral dose. So for instance, in the absence of any Trim5, it takes about this much virus to infect 10% of the cells. In the presence of human Trim5, this shifts rightward because you need a little bit more virus to infect 10% of the cells. With rhesus, it would shift even more rightward. So these are units of selection for how much restrictive potential is provided by individual variants in the course of their HIV-1 protection. And what's really nice is that even though she tested only a handful of these Trim5 variants, she found that the fold restriction upon individual testing beautifully correlated with the pooled enrichment score she found in her one-part experiment, which meant that the assay, even though it was done all together, was a beautiful kind of a reflection of what the actual constraints might be. So I want to sort of return to this waterfall plot, which I've already shown you, because one of the kind of really surprising things to us was we made random mutations in the V1 loop, hoping that some of them would be better, some of them were worse. And actually, to our surprise, we found that nearly 60% of these random variants were actually better than wild-type human Trim5, which meant that for sort of unfortunate circumstances, we were sitting in this valley, but we could very easily go up a peak where we would be actually protected with just a single amino acid change. Several of these changes are more than 15-fold more potent, which means that that would be biologically meaningful protection. So it was not the case that helpful mutations were rare here. So we became very interested in understanding what's the biochemical basis for this, because this seems like human Trim5 should be doing a lot better. And so to do that, we basically modeled our restrictors on this kind of 2x2 plot here, where the rows indicate each position in the V1 loop. The arrowheads just indicate those rapidly evolving positions that I told you about. And each column refers to mutations to a particular amino acid, with each of the 20 amino acids represented and the stop codons, because we cannot discriminate that in our mutational strategy. And so what would you expect here? The bluer you are, the more restrictive you are. So maybe this looks like a little bit of a Jackson Pollock painting, where things are kind of idiosyncratically distributed over the entire sort of diagram. It's decidedly not a Jackson Pollock painting, certainly not that he probably made any money off. And that's because we have these very row-like and column-like structures in our grafting. So the two that are most obvious is that human Trim5 has these double arginines at position 332 and 335. It turns out that any amino acid other than an arginine or a lysine would be protective. So we are sort of like doubly unfortunate with having these two arginines, whereas really we would be better off with anything else at these positions. And really underscoring that, you can also see that at any position, a mutation to an acidic residue, a D or an E, glutamic or aspartic acid, would be actually protective. So essentially, it turns out that charge appears to be really a critical determinant. It is by far not the only determinant. In particular, I draw your attention to this absolutely conserved glycine residue that is absolutely conserved across all Trim5s, across all mammals, and yet glycine is one of the worst positions, worst residues to have at this position in terms of protection. But the reason I'm not going to focus on glycine today is because charge can explain 90% of what we see on this plot. So just to sort of highlight that in aggregate terms, wild-type V1 loop has a net charge of +2. If you make a stop codon, you make it worse. If you make the V1 loop more positively charged, you make it worse. But as you make the V1 loop less and less positively charged, you get more and more protective. So even though we've known about Trim5 for nearly 20 years now, it turns out that the mutational strategy allowed us to actually predict that, in fact, the key determinant about whether Trim5 is going to be protective or not against HIV-1 is a positive charge repulsion between the V1 loop of Trim5 and a presumed positively charged V1 loop in the HIV-1 capsid. And Jeanette is now doing similar experiments on the capsid side in her own lab to basically fully kind of formulate this opinion. So again, returning to this glass half full, glass half empty kind of hypothesis, 57% of random mutations can confer protection. So this is an indication that winning solutions are not rare. So what we saw when we assessed primate Trim5s was actually a very, very small tip of the iceberg, of a very giant iceberg of possible random mutations. So next she turned her attention to, okay, what about Trim5 that is already in a winning position? Is that a mutationally fragile or a mutationally resilient solution? In other words, if you guys are skiers in the audience, are you on a black diamond ski slope where every mutation is a perilous kind of ride down the fitness landscape? Or are you one of those bunny slopes where you learn how to ski, where everything is quite gentle and unlikely to break any bones here? So to do this, she did a reciprocal version of the experiment, this time focusing on rhesus Trim5, which we already know is very good at restricting HIV-1, and now mutating each of the V1 residues and doing the same infection with HIV-1 GFP. But this time, remember, we started off with something that was already protective against HIV-1. So now we are interested in the green cells, because these are variants that have lost their protection. So it's the opposite of the assay that we did earlier, where we didn't want the green cells. Now we want the green cells because these are the loss of function mutations that we've introduced into rhesus. So again, if rhesus was very mutationally fragile, we would predict that most of the random mutations we made in the V1 loop would be a loss of function. To our surprise, there were very few loss of function mutations. In fact, the only mutations we found that were in this left-hand side enriched for the non-restrictors were the Trim5 variants that we had introduced stop codons into, so which means we had made truncation mutations in the Trim5. Most of the rhesus Trim5 variants were just as good as the wild-type rhesus. In fact, 51% of those missense mutations were not statistically distinguishable from wild-type rhesus, which means that amazingly, even though this is the primary ligand interaction interface we think for rhesus and HIV-1, it's mutationally resilient enough that it can tolerate mutations without losing its base interaction with the HIV-1 capsid. Is this a generally mutationally resilient phenomenon, or is this something that has an evolutionarily selected property on things like rhesus Trim5? We thought we can actually ask that question by asking, what if we were to engineer new restriction and then ask whether that is also mutationally resilient? And we can do that because remember I showed you that a single amino acid change from rhesus into human Trim5 can provide you substantial restriction. So Jeanette did yet another deputational scan of this variant of human Trim5, where this particular position has been changed from an arginine to a proline, removing one of those arginines that I told you are so bad for restriction. And when she did that, she found once again that this newly engineered Trim5 is just as resilient. So once you go up the fitness landscape, it's not inevitable that the next mutation is going to drive you down the mutational landscape. So this is a pretty interesting property that has been grafted into the V1 loop, we think, because of its interactions with rapidly changing viral pathogens over millennia. So the two sort of questions that she wanted to ask, how rare are winning solutions? That was a surprise. They're not rare at all. In fact, evolution turned out to be misleading in terms of the evolutionary landscape that was permissible, and winning solutions turned out to be quite stable and not at all mutationally fragile. I just want to contrast what she has found here with what others have found when they've done deputational scanning with protein-protein interaction interfaces. In particular, when you come to ligand binding interactions, what people have found is that as you get closer and closer to the ligand binding domain, you get more and more devastating effects of single mutations. It is very much like a black diamond ski slope where you have almost a lock and key type mechanism where any mutations basically break the combination lock you need for the ligand interaction. In contrast to this, we have these rolling hill-like landscapes that are explored by antiviral ligand binding domains, which you can see would be very hugely beneficial for antiviral proteins to actually have, because you're basically playing these affinity-based arms races with changing viral pathogens. So this is one of those regimes where it's easy to make a restriction happen, but hard to break the restrictions once you get, which means that you can generate standing variation without a huge cost to your standing sort of restriction profile. And these standing variants are likely to be beneficial when you encounter a new variant or a new pathogen entirely. So what I haven't told you so far, but I will right now, is that there's a reason why we think that antiviral proteins like Trim5 have this very unusual rolling hill-like landscape, which is that the V1 loop, even in structural analysis, is basically unstructured. What that means is that it's really an ensemble of many different structural states which we cannot really capture in one particular structure, unlike the rest of Trim5, which is actually quite nicely structured. You can imagine that in this situation, it's actually one of those subset of those ensemble states is probably capable of interacting with the HIV-1 capsid, and that's where we get the specificity from. However, no single mutation is going to completely break this ensemble of states. In fact, the very few mutations that did break this ensemble of states in rhesus Trim5 happen to be things where we had introduced prolines, which those of you who are sort of biochemically inclined can appreciate would be a bad thing to introduce into an unstructured loop in order to sort of impose structure into that. This should be very familiar to those of you who remember your immunology courses, because this is exactly what happens in the course of sort of affinity maturation for antibodies, where antigen recognition sites start off in this ensemble of unstructured states. And as you get more and more mature and you sort of get less and less diverse in your epitope recognition, you get more and more structured and defined. But really, you start off in these ensemble states, which gives you the maximum ability to sort of diverge into these multiple kind of valleys of affinity maturation. In fact, Trim5 is not the only protein that we see this in. I'm going to sort of switch to our second vignette now and focus on another antiviral gene called MDXA, just to make a different point, where once again, the hero of the story, if you will, happens to be this unstructured loop called loop L4, which is the primary determinant for affinity for a completely different class of viruses as well. So the disordered loops really provide the evolutionary flexibility that allows these antiviral proteins to keep pace with changing viral pathogens. So what is MXA? So just like Trim5, it's an interferon trigger gene, which is one of those many alarm genes that you turn on when you sense that some viral pathogen is taking over your body. MXA happens to be a very close relative in evolutionary terms of a protein called dynamin, which is actually a protein that is important for mediating endocytosis and the final step of pinching off vesicles as they re-enter the cytoplasm. Unlike dynamin, which is essential for cell biology, MXA is not essential, and it's actually dedicated to a role in antiviral immunity. People in the immunology world really revere MXA because it's the very first innate antiviral protein to be discovered in mammals. It was discovered because of a happy accident, let's call it that, of mouse breeding, where most laboratory strains of mice happen to be deleted for the MX locus. This was discovered many years later, which makes these strains exquisitely susceptible to influenza, both human and bird influenza. Most strains are dead within 4 days of infection. This is like a pretty significant infection. Whereas if you were to restore the mouse MX1 gene or even bring in the human MXA gene, which is the human equivalent, you can completely protect these mice. And you can do so even in mice that are mutated for the interferon receptor, which means that now you don't have access to those 200 genes that protect you against viruses. You only have access to one gene, MXA, and that is sufficient to protect you long enough for the adaptive immune system to kick in and protect you. So remember, like the job for Trim5 and for MXA is not to cleanse the body of the particular viral infection. It's just to delay the viral infection long enough for the adaptive immune system to kick in, educate itself, and then cleanse the body of the infection. That's really the primary job of innate immunity in vertebrates. Okay, so what is MXA acting against? Well, I told you it already works against influenza, and subsequent studies showed that it actually works by recognizing the nucleoprotein of influenza, which is the protein that acts almost like a histone to wrap around the RNA and mediate both transcription and replication of this RNA virus. So I'm going to it works a lot like Trim5, so I'm actually going to just go through this sort of sense of deja vu. Patrick Mitchell, a former graduate student in the lab, really focused on MXA because despite MXA's discovery nearly 4 decades ago, we had no idea what MXA is using to interact with the NP protein. Many years of biochemistry and virology had failed to identify the specificity domains. So once again, we used this evolution-guided approach where we realized that there were really 2 hotspots for positive selection, and one of them happened to be this unstructured loop called loop L4, which I've drawn in here with my artist rendition, which had the majority of positively selected sites in this. And we wondered whether this was, in fact, the missing specificity domain that allowed MXA to bind the nucleoproteins of influenza. And indeed, Patrick showed that with an influenza relative called tegodovirus, where in the absence of MXA, you have 100% infection. Human MXA is very, very good. In fact, it's still the very best MXA variant we found against tegodovirus. We have no idea why, because this is a virus that infects mice but does not infect humans. Human MXA, strong protection, so it's the opposite of Trim5. African green monkey is not protected. These are 90% identical at the amino acid level, and yet one is completely protected, the other is completely not protected. And just like what you can see from the Trim5 story, swapping in the most rapidly evolving parts of the loop L4 region between these 2 backbones is able to confer protection onto African green monkey. And yet again, a single amino acid residue out of the 5 rapidly evolving residues in loop L4 is able to convert what was a losing situation with African green monkey MXA into essentially a completely protected situation, human-like. So just to reiterate that we are able to use evolution-guided approach to identify loop L4. It does mediate the target recognition, and single residue changes can drastically alter the antiviral activity here. So up to this point, everything I've told you has been a retrospective view, which is basically I'm looking backwards in evolution. I'm trying to decipher, reading the tea leaves of where the changes have occurred, and decipher what the interaction interfaces might be. And I was perfectly happy to do this for the remainder of my career. But it's a nice thing to have graduate students because they come in as like, yeah, but that's been done before. I want to do something else, which is, what are you going to use this for? I was like, well, I'm going to use this to keep my job. That's like a important task to begin. She said, no, because you've learned all of this stuff from evolution. can you use this to make sort of a more sort of designer specificity into an antiviral protein that doesn't have it? This is pre-COVID, so we weren't even thinking about SARS-CoV, but that would be, of course, very useful. So we wondered, can we use the same playbook of positive selection identified through this retrospective analysis to now go after new specificities in the laboratory to actually elicit that? And what is the landscape of that new adaptive landscape, which might be revealed from these experiments? So this is what Rosanna TJ decided to do. She again came to my lab following up on Patrick's work, and she said, I'm really interested in loopL4, but I'm not interested in the 20-odd primate sequences that you've sequenced, because that's a very small subset of the possible combinatorial space of those 5 loopL4 residues. I want to mutate each of those residues, but not one at a time in the deep mutational scanning approach, but combinatorially, because I want to explore what are the possible states that might be revealed by this combinatorial mutagenesis approach. So I said, this sounds like a good idea. We should do this. This was many years ago. This would now be like a phone call to Twist Biosciences away, but unfortunately, that was not Rosanna's good luck. So she cloned these through PCR mutagenesis, selected variants, and assessed them individually in triplicate for functionality. And she did about 1,000 variants. This is a lot of work. I'm not going to sort of deny that this was a lot of effort that she did in this part. And it was very disappointing. So what she found was that 95% of the variants were actually worse than wild-type MXA. So even though we had started with human MXA, which is very good, and we had only messed with the rapidly evolving positions, it turns out you can still break MXA quite easily, even just by that messing around. And about 5% were about as good as wild-type MXA. And then we had a few variants that were slightly better even than wild-type MXA. And so this was like one of those deeply depressing series of lab meetings where you sort of get these accusatory looks about graduate students about how you should have really told me better things to do. And then we sort of realized after that had ended that we should look at the variants that did have some activity. And it turns out that all of those variants actually have this residue restriction, which is that they need to have a phenylalanine, a tyrosine, or a tryptophan at residue 561, which those of you remember was exactly the same residue that Patrick had actually mutated from human into African green monkey to confer protection. So the other moral of the story is we should definitely reread our own papers before embarking on the next study. So my most successful contribution to this project was to convince Rosanna to not quit grad school and redo the screen again, but guided by this approach where she had already found that phenylalanine is very good. Turns out tyrosine is even better. And tryptophan can work, but it's actually not very good compared to the other 2 hydrophobic residues. So we decided we're going to fix it at phenylalanine, which is what the human variant is anyway. And just to sweeten the pot a little bit, I had a rotation student, Emily Shea, join Rosanna. And we said we're only going to do about 250 variants, right? Because we're going to test them individually. And this could have gone really badly, but luckily for me, it did not. So now 70% of the variants were as good or better than human MXA because we had now honored the requirement of a hydrophobic aromatic residue at the critical position. And we still have about 30% of variants that are worse than what we'd expect. And I would be really interested in answering why that is. But of course, we never did that because we realized that we had now nearly 24 variants that was significantly better than wild-type human MXA, which is the very best natural restrictor of tegodovirus that we have in our arsenal. So this is that data shown in triplicate again. Here's a catalytically dead version of MXA that we use as a control normalized to 1. Wild-type human MXA at this level of expression is 10-fold protection, really, really good protection. Here's our initial 5-site screen. And here is now our 4-site screen where we have fixed the phenylalanine. So I just want you to appreciate what we've done here. We arrived at loopL4 by a retrospective analysis that identified these positions. We have now just allowed those positions, 4 of those positions to mutate. And we now have 10 to 20-fold better protective versions of MXA just by taking this evolutionary sort of handbook and just allowing molecular biology in the lab to tell us about allowing us to screen more variants of that type. I just want to point out, focusing on the 3 very best super restrictors we have on the right here, that these super restrictor variants are better at all levels of expression. So they outperform wild type MXA, where I'm just going to sound like a broken record, very best natural restrictor, and these variants we have in the lab can outperform them significantly at all levels of expression. And unsurprisingly, but still reassuringly, the reason they outperform this is because of higher avidity of interaction with the nucleoprotein of tegodovirus. So here you can see that wild-type MXA has the really nice ability to co-IP with the NP protein during virally infected cells, but each of the super restrictors is much more avid binder of the NP protein, and that makes it a much more avid restrictor of the tegodovirus itself. So let's return back to this sort of diagram where we have now the 5-site and 4-site mutations that are beneficial. This is the point in the talk where I often get asked, now you could use deep learning to actually like figure out like what is the secret here? Like what could we do? And that's the point in the talk where I tell you, this is not a representative set. This is literally all the variants we found at the end of Rosanna's set. We could certainly find more, but that would require her to come back to grad school, which is not happening. So we use my version of shallow learning, which is the PI stares at these variants for like a very long time and like allows patterns to emerge. And actually this did work in this case because we realized that the Q residue, the glutamate at the first position, was actually kind of overrepresented here, in spite of the fact that glutamate itself is not that well represented in the genetic code. And so we said, aha, the glutamate at this position, position 540, must be critical. So just to remind you again, here's the catalytically dead version, wild-type MXA with the 5 residues as those rapidly evolving positions. A G to Q mutation is a small bump in restriction, but actually if you take each of the super restrictors that already have a Q, reverting to a wild-type version, you lose the super restriction. So this is our first evidence that residue 540, in addition to residue 561, is playing a very critical role. That prompted us to ask, okay, what about other amino acids at this position? Maybe we should mutate them to all of the amino acids that are present. So we did that, and actually turns out that proline is terrible. Again, unstructured loop, putting a proline very bad, so we're not going to kind of dwell on that. That turns out to be a universally true thing. But you can see that you can go from tryptophan on this end to alanine on this end, and that's a 10-fold difference in restriction. That's the difference between, protection and no protection, if you will. And that's just happening with a single amino acid residue. And we stared very hard at this to understand, are there any biochemical patterns, et cetera? And nothing really became obvious until, again, shallow learning came to the rescue, because it turns out that valine and threonine are really bad in the wild-type context. But I remembered that valine and threonine were actually present in 2 of our best super restrictors. So suddenly visions of converting those valines and threonines to alanine and getting a super duper restrictor, like, wow, that would make a really great final figure for a paper. And so that's what I encouraged Rosanna to do. Let's take the wild-type residue in which a valine to an alanine mutation is this dramatic difference in restriction. Let's do the same for the super restrictor that already had a valine, and maybe we can even bump up the restriction even more. Of course, the opposite happened. So the same mutation in a different context, remember these are only different in 2 amino acids of the loop L4 compared to the rest of the 600 amino acid MXA protein, have completely opposite effects on restriction profile, just again reflecting the unpredictable nature of the ensemble of these different states, also really highlighting that this kind of epistasis means that we were really lucky that we did combinatorial mutagenesis rather than single residue scanning, because we were a totally discarded valine, but that loss of function mutation actually permitted the gain of function subsequent mutations to actually occur. So you had to go through the valley to get to the peak that was actually afforded. So just to sort of summarize what I've told you so far, we started with this approach where we looked at evolution and identified loop L4 as the critical determinant. Turns out not just for tegodovirus, but also influenza. And now Rosanna has taken this combinatorial mutation approach where she's mutated all of the residues except 1, and she's now found super restrictors of tegodovirus. I can guarantee you that most of you have never heard of tegodovirus or wish you had never heard of tegodovirus, but you really all care about influenza virus. So suddenly visions of grant funding began dancing in my head. I said, oh my God, what if you've discovered super restrictors of influenza virus? Wouldn't that be amazing? And maybe that's what the whole story is going to be about. And that's when Rosanna tested many of her super restrictors against Tagotovirus on the x-axis, against the H5N1 strain of influenza virus on the y-axis. Here's the wild-type MMXA. And you'll realize what became apparent to us over like weeks of doing this, that you could get really good at Tagotovirus, but when you did so, you got worse and worse against influenza virus. So even though we are messing with this very small combinatorial mutation landscape of the V1 loop, there is this breadth specificity trade-off where the wild-type version that we have in our genome is really the best jack-of-all-trades version against both Tagotovirus and influenza virus. You can get better, but you often do so at the expense of the breadth of restriction that MXA can provide. So undaunted by this, a new graduate student in the lab, Rachel Geiger, said, well, I want to do this approach, but this time against the, the influenza viruses, because that's the ones that I care about. And long story short, this approach works against influenza viruses, but we find completely different variants. This time, we made the judicious choice of allowing all 3 hydrophobic aromatic residues to occur in this combinatorial mutation library. And that turned out to be a very judicious choice, because what she discovered was that at residue 561, which is the critical residue, the tryptophan residue is overrepresented among the influenza superrestrictors, and the tyrosine residue is underrepresented among the influenza superrestrictors. In fact, this is clearly apparent by taking just one variant where we basically mutated just this residue from a tryptophan, which is super restrictive, and a phenylalanine or a tyrosine, which loses that super restriction. And if you'll recall, this is exactly the opposite of what she found with the hydrophobic residue preference at residue 561 for Tagotovirus. So it actually turns out that this breadth specificity trade-off often actually just comes down to which hydrophobic residue do you have fixed or not fixed at residue 561. If it's a tryptophan, you're much more likely to be an influenza restrictor. If it's a tyrosine, you're much more likely to be a Tagotovirus restrictor. If you're phenylalanine, it turns out you still have some play. You can be both, and we're happy to explore that. So I just want to tell you that the jury is still out whether we can use this approach for super restrictors or influenza virus. We feel confident that we'll be able to do that. And the next step, of course, is to go after pandemic strains of influenza that basically have escaped human MXA and ask, can we use the same approach to bring them back within the restrictive manifold of MXA restriction? So hopefully I've convinced you today that disordered loops, even though they're kind of really messy from a structural standpoint, are really an important tool for antiviral proteins. They provide both the evolutionary flexibility and the adaptive potential that allows antiviral proteins, even slowly evolving. Most of what I told you today are one-on-one arms races between antiviral proteins and rapidly evolving viruses. And these red queen conflicts can nevertheless be quite successful. I just want to close by acknowledging the people who did the work. All of this work is actually the consequence of a very influential coffee that I had 20 years ago when I started my lab with my senior colleague, Michael Emmerman. I hope it was not totally obvious today in my talk, but I'm not a virologist. I'm an evolutionary biologist by training, and all of this is a beautiful symbiosis between a virology-driven lab and a genomics and evolution-driven lab. So we've really trained a lot of students, and there are students in my lab doing experiments which I have absolutely no training in doing and no biosafety ability to actually train them, but Michael does, and that's good. I also want to thank Jeanette for leading all of the Trim5 work and actually like retracing our steps on an important study and finding even more insights, and Rosanna and Rachel for the super restrictor. Thank you very much.
Hello, thank you very much for a very beautiful talk. So I have like 2 quick questions. The first one about Trim5. Do you think the fact that humans are stuck with this bad version of Trim5 is likely a result that it's bad for HIV but good for other retroviruses?
Excellent question. I have a whole different part of the talk where it talks about why did we fix those double arginines. The short answer is double arginines seem to be bad for HIV and for SIV, all the SIVs we've tested. So we are still on the hunt for, this was clearly driven by adaptive mutation, which means that this was beneficial when it occurred, the double arginines, but we still don't have what was the driving force for that evolution. Whether it was a virus or whether it was to diminish some sort of autoimmune response, we don't know.
Thank you. And then second question related to that. So if there seems to be some sort of mutual exclusivity, like one variant is good for one virus, another for another virus, others genes like frequently duplicated in evolution, because that would be very beneficial for them to be evolving separately.
This is an excellent question. Even if there was no specificity, it would be massively advantageous to have allelic heterogeneity as well as gene duplication, a heterozygote advantage at each of these. That is true for Trim5, and it's actually true for most self-respecting antiviral genes among mammals. It is not true for MXA, and we do not know why. I would completely agree with your prediction that MXA should duplicate, and basically each version should specialize for a different virus, because then it doesn't have a trade-off or a compromise. We don't understand the nature of the trade-off. There's another student in the lab who's very interested in why that trade-off is occurring. Every mammal, despite multiple rounds of duplication, has two MXs, one that works in the cytoplasm and one that works in the nucleus. They work on completely different viruses, but there are not cases where there are multiple MX paralogs within the same. And so we are actively considering the prospect that because MX acts as an oligomer, having paralogs in the same compartment runs the risk of poisoning the oligomerization between them, and that might lose the activity. And that's something we are actively studying right now.
Thank you.
Thank you so much. Very interesting talk. My question is about the kind of super restrictors and the trade-off you mentioned between the Togotovirus and influenza. When you look at like species where only, like host species where only one of those virus families has circulated, do you see kind of a preference for one or the other kind of mutational status of the super restrictors?
Yeah, that's an excellent question. So we don't have the ability to make that judgment quite so well because, as I pointed out, the Togotovirus is not something that is present in human populations. It's something that is endemic in European populations of mus musculus. But that doesn't mean that it wasn't able to actually provide some protection. So it's really like if you were successful, you probably don't have the virus because you're protected from zoonosis. So that kind of ascertainment bias is really hard for us to reconcile. I will also point out that influenza viruses, at least in their normal course of infection, probably didn't drive the majority of the evolution we've seen because, again, that was in the African diaspora. So there are viruses that have driven this, but we're using Togotovirus and influenza almost like surrogates for what those viruses might have been. Yeah.
Thank you.
Yeah, so it seemed like for Trim5 there was no evolutionary pressure with regard to HIV-1, probably because the history was too short. For MXA, obviously there was a lot of evolutionary pressure, and you hinted at the possibility that there could be versions of MXA that would treat influenza and the other viruses perhaps equally well and better than the wild-type version. Have you looked at whether perhaps any of those mutations that would be better from an antiviral perspective would lead to responses in the organism that would be disadvantageous?
Some sort of toxic effect of their...
toxic effects or trigger adaptive immune responses or whatever, because that seems to be another point.
Yes, so I will completely agree with that. That's actually, by the way, true of Trim5 as well, where Trim5 is an E3 ubiquitin ligase with a rapidly evolving ligand specificity domain, and you can imagine that some of the adaptive landscape is going to be restricted because of the chances of an interaction with the self-protein. All of our experiments are done in cell lines, so we have not actually manipulated. Each organismal infection is like a significant investment, so we've not, and assessing which of the variants that didn't work we want to test is more problematic. I will say, though, that everything in our assay, we did not actually see any overt toxicity with any of the variants. So both in the V1 loop of Trim5 and in the MXA, where we are able to assess, because we tested each of these individually, nearly 2,000 variants, if there was a toxicity variant effect, we would have noticed that. They all appear to be expressed well, and they are all capable of oligomerization. That's still only about 1/1,000,000th of the landscape that is afforded. That doesn't mean there's something not afforded. I just also want to make a small correction. Actually, with MXA, we don't know what actually drove it. With Trim5, we have a much better sense of what drove it because the human Trim5 is very, very good at restricting entropic murine leukemia virus, which is the closest surrogate for many of the endogenous retroviruses that we have integrated in our genome. And almost certainly, I don't think there's any ambiguity that a lot of the changes for Trim5 were driven by those arms races with those endogenous retroviruses.
Thank you. It seems to me that the best strategy for a virus would be to not only evolve to avoid the recognition by the innate immunity proteins, but to also evolve such that it became more similar to a host protein, to almost take a host protein hostage so that if that innate immunity protein evolves to chase it and hit it in the future, it's going to have to somehow like do a second step to avoid hurting another host protein. So is there any evidence that something like that exists, or?
Well, I'm smiling because Tom Dever is in the audience, and his lab has, along with my lab, studied many examples of viral mimicry, exactly what you're saying, where viruses will steal host genes and use them as decoys to essentially prevent antiviral proteins. Often they'll steal host genes which are substrates of antiviral proteins and make versions that are capable of being bound by the antiviral proteins but not capable of being catalyzed, which basically like takes them out of the picture. This has happened. This is a very successful strategy that many viruses that have the ability to have large genomes, like double-stranded DNA viruses, have employed very, very successfully in the past. There's no evidence for that for MXA or Trim5, but in theory, your point is well taken that mimicry is a very, very good evolutionary strategy in general.
There are online questions, Chris?
There is one. We're running a
Should we
bit
do
late.
the in-person question first?
Maybe why don't you
Okay.
Hello. It was so interesting because you focus on the flexible loops, because those are very important. It was here in my lab, we showed first the role of flexible loops in protein structures with lactose synthase, glycosyl transferase. Now what I want to ask you a specific question is that you have a glycine residues attached, and the glycine motion conformation, you must keep or analyze the conformation at that region and the dynamics of the conformation. That's very important. Glycine will change.
Glycine and proline are both interesting at both ends of the spectrum. Exactly.
Yes. Yeah. That study is very important.
Yeah, I should point out that we have not actually done much in terms of the conformational state, in part because we think about, with MXA, we've been thinking more that it's almost like an unstructured loop that becomes an induced fit when it's bound, and there are no co-structures of the MXA with the NP protein, in part because the avidity was not strong enough for a co-crystal structure. We are hoping that our super restrictors will solve that because we have 20-fold more avid structures, and we are actively working with colleagues. We've been told that the conformational status is something that is really the purview of NMR, which is, this is far too big for that. But we're hoping that even cryo-EM studies might actually reveal a little bit more about those conformational states. Thanks for the question.
Maybe one, if you don't. It's part two of this one question from our colleague in the library, National Library of Medicine. Could the story about the two arginines, rather than being an unfortunate coincidence, reflect adaptation to HIV to humans?
I think I answered this question.
That's what that was?
Yeah, it's not about adaptation to HIV or really to any SIVs. We've tested nearly a dozen SIVs that could have possibly jumped into humans, but maybe were protected because we had the double arginines. But it is certainly adaptive. There's no question. This is an unfortunate coincidence only with respect to HIV susceptibility, but it was not an unfortunate coincidence because both of these were deliberately occurred and deliberately selected for in the context that they occurred. We just don't know, since this happened in the common ancestor of chimps, bonobos, gorillas, and humans, we just don't know what that agent was, whether it was a viral agent or an autoimmune agent. That's one of the problems, is that making a contemporaneous argument for what drove this requires us to have a pretty good sense of what the viruses were. With retroviruses, we have a very good sense of that because many of them integrated in our genome, but things like HIV don't integrate because they're not germline integration events. So we have half the mirror image of the story. We are not able to, we know it's adaptation, but we don't know what caused it.
Okay.
All right, thank you very much for coming.
Thank you so much.